I gotta ask, what was up with the pig that you showed?
I think that was it green? I don't know. It was some genetically engineered.
Okay.
I think it expressed green fluorescent protein or something.
I see. Okay.
Yeah.
Yeah, for cancer treatment, is there any kind of bridge species before you go from mice to clinical?
Yeah, well, so in order for anything to go in patients, it's got to be tested at least 2 animals. So it could be dogs and mice or something. But I mean, with chimp research kind of on its way out, it's kind of not really feasible either. I mean, you have to kind of bridge this. I mean, the reason the mice are used so much is they're small, they're cheap, fast gestation times. You can manipulate their genome. So that's kind of the problem with going to the next one. Just the time and the cost and the. So one of the things with the pigs that we think is going to be kind of cool. You can get, they make mini pigs now. You know that only this big, and they cost. If you put them on, if you have them on a farm, so a biomedical farm, it's only $3 a day for a pig to keep a pig alive, right? It's $1 something a cage for mice. And I think the information you can get from the pig is vastly more superior than you can get from a cage of mice. Like we can house mice on HSC campus in the bottom basement of the Cardiovascular Institute for $11 a day for mini pigs. So it's actually not astronomically expensive if some of the things that we're proposing can get done.
Well, thanks for your presentation. I just have one question because I am interested in lung cancer.
Yeah.
Is there 2 types of lung cancer, small cell and non-small cell? Your way that you are using here, it work for both or just one type?
So it depends. So I mean, even within non-small cell, there's 3 types of non-small cell, right? Adeno, squamous, so and large cell. But it depends. A lot of times, like in the animal models, you can induce small cell or non-small cell based on what genetic factors you put in. So. And then that depends on like direct infection with viruses or with transgenic animals. It depends on the combination of mutations.
Also, okay. Also, your way when you are doing it, you can control that the tumor can appear only in right lung or left lung, or you don't have this.
The only way you do that is there are techniques where you can orthotopically inject into one lung. So basically, you just poke the needle right into the lung, and usually the cells are mixed with matrigel or something, and you can do that. And actually, Jun Yan over on the other campus has published a paper using that model.
So I was just curious, though, your animal models, are you actually breeding them here, or do you pay a commercial laboratory?
So the breeding in our building, CTRB, I mean, we have hundreds of cages. I probably have over 100 cages of animals. And so, but most of the animals, so if we want to, they are commercially available, almost everything I talked about. So if you wanted to get them in and start your own colony, you could just pay a few $100 and get a breeding pair. So like I said, it's about $1 a day per cage. And you put 5 animals in a cage.
What's the time scale typical for developing a model?
Developing it from, and which kind of model? I mean,
like say the chimeric model that you're.
Well, some of those I could start today and have tumors in 2 weeks. It depends. It depends. So if you want to do a chimeric where you're injecting virus, is the virus available? Do you have to clone the gene in, and then make the virus, and then what's the latency? Is it actually going to give tumors? So there's some variables there. If you wanted to make a genetically engineered model, it could take a year from initiating the project to getting mice. So like our lung cancer model that we've used before, it takes 6 to 8 months after they're born before they get lung cancers. And that's just driven by mutant KRAS. So you could add things on top of that to then drive it quicker.
So related to that, with the mice, a lot of times you dump in a whole bunch of cancer cells and you've got tumors within a week or less sometimes.
Yeah.
With the pigs, do you anticipate that would be a longer time scale for tumor development? I know
Not
obviously
necessarily.
humans it takes longer anyway.
Right. Well, yeah, so again, it depends. Like in humans, you're starting with one cell that has the acquired mutations that now divides and turns into a tumor, whereas like you said, in the mouse, you could just take 1 million tumor cells and crank it in, and then you got your model ready in a week. Pigs is similar. There are skid pigs actually that are available that you can use, and you can inject human cells under the ear of the ear skin, and they'll develop little subcutaneous tumors. The thing about genetically engineered, there's a lot of genetically engineered pigs that are coming available right now. And so there are pigs that have loss of P53, for example. There's a pig that has mutant P53 and mutant KRAS. And those have this pre-inducible transgene in every cell of its body. And the group, what they did is they took adenovirus expressing CRE, and they crammed like 10 to the 10 particles, like just started injecting it in different places. And they turned in within 20 or 30 days, they had these masses of tumors that were driven by mutant P53 and mutant KRAS. So those are non-physiologically relevant scenarios, but the other thing too is that typically if you take cells from a pig and you put it in culture, it takes 6 to 7 mutations to transform them in culture. Whereas a mouse cell, it takes 2 to transform them in culture. A human, it takes 7 to 10. So again, from a genetic standpoint and the checks and balances, a pig is closer than a human. I mean, a pig is closer than a mouse by far.
Well, you mentioned trying to get the pig model up and going. Just curious, does UofL, I know what the RRC facilities are. How are you going to house this, I mean, the much larger animal if you're talking about doing hundreds of animals?
Well, it wouldn't be hundreds. So I think over in the CII, in the basement of the CII, they have the facilities, and we already have a protocol in place to start working with pigs now. But I think they can only hold like 12 at a time or something. But that's, I think, where you would have to get with. There's this National Swine Research and Resource Facility that's in Illinois, I think. Anyway, that they have farms where you can basically put the pigs on the farms for $3 a day.
So if you got like a center grant to start this, are you talking about building a new building?
I mean, if you gave me $50 million right now, I would definitely build a facility out on the Shelbyhurst campus probably and start to house. I'd start running clinical trials, because the cool thing I didn't talk about is it turns out these immunotherapies that are used in human patients actually work on the pig cells. They don't work on mouse cells. But that's how similar the proteins in pigs are in humans, is that you can actually use the same drugs that are targeting proteins in humans and in pigs. So you could imagine basically a preclinical trial with the exact same drugs at the exact same doses. Because the other problem with these models, even with the cisplatin that I talked about, is we're injecting everything bolus. You know, if you're a cancer patient, you're going to the clinic and you're sitting there for 2 hours getting your cisplatin IV in the context of up to 1 liter of saline. So that's completely different. So the blood, the peak maximum that you get in the blood, a bolus versus 2 hours IV drip is completely different too. And so you can hook a pig up to an IV bag and give that pig IV drip for 3 hours. So again, that's the next step of relevancy that we want to hit, is being able to give the drugs exactly like they're given in the clinic. Yeah.
So did you try to use your model in something rather than mice, like rat, rabbit?
We have not. I mean, just we've just done mice so far.
Because I am asking this question because when we try to detect the tumor using MRI, the lung of mice is very small.
Yeah.
So if we try it in some big animal models, will be very easy for us to wait until the tumors become appear.
My rebuttal to that would be the pigs again. Because actually in the grants and in the protocols and stuff that we're writing now, we're saying we're going to induce lung cancers in the pigs, either small cell or non-small cell. And then at certain times, we're actually going to put the pig in the MRI and follow. So, and that would be his most. And the other beauty of that is you can imagine surgical oncologists in training working on a pig would be like as close as you get to getting in working on a human. So, there's just some of our other ideas for that.
All right, well, everyone, thank you so much for attending today. If you're interested